A very good afternoon, everyone. Let's crack CSIR net together. Welcome in today's class. I'm your educator Farida Bandookwala and I'm pursuing biotechnology from School of Biotechnology, ABB. And I've done my BSc, MSc, and MPhil in biotechnology. I have a teaching experience of 10 years and I've qualified MPCET examination. Before beginning with the topic of today, let us have a small introduction about Unacademy. Unacademy is one of the India's largest platforms for the PG examination. If you are preparing with Unacademy, what you will get or what it benefits, you will get daily life classes so that you won't be left out. Every day you will be that today I have to do it. So daily life is a benefit. And there are structured courses like the courses they are in a very systematic manner so that you can every point thoroughly and they are well arranged as the topics in the CSIR. They are lengthy, they are vast, they are 30. So are planning to study them, they need to be very systematic in your study. Otherwise, you will wander around here and there. Syllabus should be the pattern of study should be very systematic, it should be structured, so that you can get from Unacademy. And there are live tests and quizzes which are conducted at an academy. So these live tests and quizzes can uh, breakthrough point to show you the reality whether you have learned or not. Some live tests and quizzes can be very much helpful. And you will have unlimited access towards all the data available on the platform of an academy in the category whichever you are subscribing for. So when you are going for an academy, we conduct free live classes, and these free live classes which we conduct, they are conducted platform. One of the class which is going on now, upcoming in class, kind of free. Then another place where we conduct the class is called as a special class. For special class, you need to download the app, and you can download the app by writing Unacademy app on it, and you will see the Unacademy Learning logo. Just download the app, make your account, and select the option CSIR-UGC. Good to go with the preparations. There are special classes at an academy. After this, there is this plus subscription. And what is so good about this plus subscription is the 30 well arranged systematic manner. So plus subscription gives you the um, access towards the few lectures which have been aired or previously they were live. You can go and check them out. And on the Plus platform are always some kind of crash going on or short-term course going on for the upcoming exam. So they always keep you in that motion and in that uh, what to say in that kind of thought process that yes, you are preparing for the examination. So this plus subscription, you pay a certain amount for this plus subscription, and it is quite pocket friendly. You don't have to spend a lot. So spend a very little amount and access for the plus subscription. So for 12 months, you need to pay around 10,000 for 12 months. So for it goes again very down, that is 50,000. And in this amount also, if you are using my referral code that is FP1, you'll be getting a 10%. Don't forget to use my referral code FP21 for getting this extra 10% discount. This was a small introduction about Unacademy. Now we'll be talking about Hardy-Weinberg's law. Hardy-Weinberg's law. Now this Hardy-Weinberg's law is called as law, can be called as principle, can be called as theory, or it can be called as any word which can be used. So all are one and the same thing. So what does this Hardy-Weinberg law actually tells you about? Hardy-Weinberg's law tells you about the frequency of population. Tells you about frequency of allele population. Tells you about frequency of allele population. This is so. If you study allele in population, it is said that. So we'll take another place. If you study allele, single allele in population, study a allele population, it is said that frequency of the allele or the genotype, it is going to remain constant if population is not subjected to any kind of evolutionary force. So the frequency, it is said the frequency of allele or genotype will remain constant is going to remain constant if that population is not subjected to any evolutionary force. This is the statement of Hardy-Weinberg law. So if a population it is not subjected to any kind of evolution force, the allele or the genotype they are going to remain constant. It means there will be no kind of development in the types of allele and genotype, they are not going to change. This is the statement of Hardy-Weinberg law that the allele frequency will not change from one generation to another generation. Frequency of allele not going to change from one generation to another. This is what Hardy-Weinberg had to say. Now, to understand Hardy-Weinberg statement, we need to talk about certain things, and we need to see the numericals based on Hardy-Weinberg law, because they are the most important things which are asked in examination. In examination, they are not going to ask you tell directly about the Hardy-Weinberg law, not the statement. But the most important questions which come on Hardy-Weinberg law is the numerical part. So we are going to talk about the numerical part in the Hardy-Weinberg's law. So if we are seeing chromosome, so in chromosome there will be genes. So we are using P for dominant genes for a single kind of protein. From is for dominant and Q. Right. So in a population, what will happen? In a population, what will happen? That if you are writing P plus Q equal to 1, this is the allelic frequency. Allele frequency. Either a person will have P or either he will have Q, because he cannot have both of the things together. So in a population, the allelic frequency will always be dominant plus recessive is equal to 1. So let us write this thing, this condition. So this is the formula. The formulas are so simple that you won't even think that they are kind of formula. But you have to use these formula for understanding the language which examiner is using. The language is the important thing, not the formula. Formula is very easy to remember. You won't face any kind of challenge in remembering the form in formula, but the thing is, solve the question. Yeah. So the condition is either P or Q at one loci. Either P or Q will be there at that particular loci. And genotype frequency, the genotype frequency, what it will be, the frequency of genotype, what it will be, genotype will be either dominant dominant, recessive recessive, or it can be dominant recessive both. So the genotype frequency, if a person is homozygous, if a person is homozygous dominant for both of the genes, homozygous recessive for both of the alleles, or a person can be heterozygous for those alleles, dominant and recessive both having. This is equal to 1. So this can be one kind of condition, isn't it? So this is also our one of the formula of Hardy-Weinberg law. So this is it. Are you seeing how simple it is? And you know this p square plus q square is equal to 2pq is equal to 1. This we have got from where? So just for your information, I will explain that from where this comes. You know, this comes from plus q. Sorry, there should be a multiplication. So p plus q into p plus q equal to 1 into 1. This is the thing which is leading to this. But this was quite easy to understand in terms of genotype frequency. You people can remember it very well. Homozygous for that particular gene, heterozygous for that particular gene, and homozygous dominant or recessive for it. So that's why I just directly did that. And this one is the constant, and it is not going to vary according to the Hardy-Weinberg law because there is no evolutionary force. This is constant. This one is constant. This one is constant, and it will not vary constant. And if it is constant, it is constant according to Hardy-Weinberg law. Constant according Hardy-Weinberg law. Nothing else. It is just constant according to the Hardy-Weinberg law. None other than that. So genotype frequency we have seen, and now we will write the conditions over here. This PP is telling you about homozygous dominant condition, homozygous dominant condition, and if we are writing QQ telling us about heterozygous condition, heterozygous recessive, and if we are saying PQ is telling us about I've written heterozygous recessive, sorry, this is homozygous recessive. Homozygous recessive and PQ is heterozygous. So this we are understanding from p square plus q square plus pq is equal to 1. This we can understand from this formula. Now what are the factors of Hardy-Weinberg law? The factors. What are the factors of Hardy-Weinberg's law? So there should be no mutation. Instead of factors, can we use a word condition? Yes. Much better. Factors instead of factor, we should say condition. The condition is no mutation will occur. Then no random mating will occur. no mutation will occur, no random mating will occur, and population size should be large. Population size should be large. Population size should be large. Do not forget that the size of the population would be large as compared to when we were talking about genetic drift, the population size should be small. Here the population size should be large and there should be no sexual selection as well. No sexual selection. No sexual selection should occur. So it's not maintained or followed by any population. So this is not a very true condition. This is not maintained or followed by any population. Populations, they have this kind of thing that mutation will occur, random mating is going to occur, these all things. So what we can hypothesize is that we can calculate the allele frequency or gene frequency from Hardy-Weinberg law and how the population will behave in certain situations. So what all we can make out from Hardy-Weinberg law? So what can we get from Hardy-Weinberg's law? What all things we are going to understand. So the thing that we can understand is we can calculate allele frequency oblique gene frequency. And what else? We can also understand that how a population is going to behave in a certain situation. How can population behave in certain situation? These are the things that we can make out from Hardy-Weinberg's law. Now, as I told you in the starting only, that Hardy-Weinberg's law is a small law, and moreover, the questions which are asked from Hardy-Weinberg's law is based on the questions are based on the numericals, right? So we'll be doing the numericals. I have selected some of the numericals for understanding. So the first numerical is in a population of 1,000 individuals. Homozygous dominant or homozygous condition for AA is 360 and heterozygous condition is 480 and the remaining are 160. So based on this data, calculate the frequency of capital A allele. Now can you see the question? What all we can get from this? Let me red color. So here we can see this thing. Here we can see this thing and the remaining are 160. This also they are saying. So this remaining 160 is actually what? Small a, small a. So based on this data, we have to calculate frequency of capital A allele. This is the question. So we know, we write over here capital A is capital A, small a is 480 and small a is 160. Now we have to find out frequency of allele. So how would we calculate the frequency of allele? So if we are calculating the frequency of capital A, as they are asking in question that you have to calculate the frequency of capital A allele. So capital A allele is present here in this situation. So we can take 360 from here. And in the heterozygous condition also it is present, but it is present just half. Because it is heterozygous and it's a diploid kind of population, that's why they are writing AA. AB, because this is like diploid population. So half of capital A is present in the heterozygous condition. So half of 480 is 50. So like this, the A becomes 600 and the total population is of 1000 individuals. Can you see the total population of 1000 individuals? The frequency of allele is very easy to calculate. 600 divided by 1000, that is 0.6. This is the frequency of our capital A allele. This is how one calculates the of the allele. And if they ask you the frequency of a small a, so you will apply the formula is q to 1. So zero point 0.6 plus q. Instead of q, better to write a because they are taking a as the another allele. So in this case, the a will be 0.4. So this is how you can calculate. So it will not be this simple. It's not going to be this simple that they will be just telling you about the value of P and the value of Q and you have to calculate things. So they will always ask you in these terms that there are so many individuals and the individuals are having this frequency of this and that. So you should see what they are saying. So in a population of 1000 individuals, they are telling you the genotype, and you have to calculate the frequency of allele. The thing what they are giving is the genotype, and you will try to find out the allele. So another question also I have taken in this context, and that is about a census of birds nesting on Galapagos Island revealed that 24 of them show a rare recessive condition that affected the beak formation, and other 63 birds in this population show no beak defect. Population is in Hardy-Weinberg in equilibrium. So this is also very important thing over here. So a standard question given by a well-read person will always indicate that it is following Hardy-Weinberg equilibrium, or it will not say Hardy-Weinberg in equilibrium. They can use the language that it is in equilibrium. There is no random mating, no random mutation. There is no immigration, no immigration from the population. These all things they are going to tell you. 24 are having a recessive condition, 6 are complete all right. What is the frequency of dominant allele? And answer should be near hundreds. That is the question. Here also you have to frequency or genotype. What is given in this? So you are solving this for allele frequency. You are not solving it for genotype. And what is given over here is the genotype. So the total number of birds, if they are saying that reveal that 24 of them and other 63 were right, completely all right. So total number of birds, how many were there? Total number of birds, how many they are? Total number of birds, how many they are? Plus 20. That is total number of birds is 87. Now here they are telling you about the genotype. Here they are telling you about Q square, and this is your Q or QQ, and here they are telling you about the EP, right? So this is what they are telling you from this. Now we calculate. So recessive genotype is given, and for recessive they are saying that Q square is equal to 24 divided by 87. What you are going to get from this is 0.27. Then if you are getting 0.27, so what is Q square? That is 0.52, right? And if you are solving for P, can be Q minus 1. So the Q is 0.52. So Q minus 1, it should be 1 minus Q, right? Isn't it? Yes. So it should be 1 minus. So here the answer would be 0.47. So this is how you calculate. So yeah, this is how you calculate, how you are going to calculate the alleles, right? Now another question is, at a particular locus, frequency of a allele is 0.6, 0.6, 0.6, and for small a it is 0.4. What is the frequency of heterozygous in random meeting? Another kind of question. They are saying a is equal to zero point 0.6, b is equal to zero point 0.4, and now we need to find out p. We use the formula p square is q square plus pq equal to 1. So 0.6 square plus 0.4 square plus pq equal to 1. So on solving this, 0.36 plus 0.16 plus pq equal to 1. So q equal to 1 minus 0.5252, that is equal to 0.14. So the heterozygous will be 0.14. So this is how you are going to solve the questions of Hardy-Weinberg equilibrium. You should first of all identify what is given, and if you have understood that what is given, you can identify it as genotype that is given, or if it is the allele frequency that is given, it will be very easy for you to understand and solve your question. So try to solve as many questions as you can in Hardy-Weinberg equilibrium, and this is like one of a question which is usually asked, short question. So in the end, what we say, let's crack CSIR net UGC, CSIR UGC net. Hit the bell icon for getting the notification, and also here give a thumbs up if you like my video. Please subscribe to our channel, that is Let's Crack CSIR UGC net. And if you're going for plus subscription, use my referral code, that is FB01. So do not forget to use my referral code FB01. And if you're having any kind of doubt or confusion, you can write your query in the comment box, and you can also follow me on the Unacademy learning app by my name, that is Farida Bandookwala. You can follow my profile and you can ask me doubts and queries over there also personally. So this is it for now. We'll be meeting with another lecture of evolution this month, and maybe carbohydrate metabolism this month, and we'll be continuing our study. So this is it for now. Bye.